to CCF, and thank you to Giant Foods for having me today. What I'll be talking about is the updates from our phase 1 CD22 CAR T-cell trial and sort of where we are, but we'll start off with a little bit of a background on where we are with CAR T-cell therapies in hematologic malignancies overall. And absolutely, the Next Gen Award was tremendously helpful in helping sort of establish and get me to the next stage of my career. No disclosures, and CD22 CAR is not FDA approved. It's a phase 1 clinical trial. So in terms of making a CAR T-cell and what a CAR T-cell is, so this was mentioned very briefly in Christine Heskey's talk, but basically what we're trying to do is we're trying to take T-cells from patients and give them the recognition capabilities to identify leukemia as a target. So you take a T-cell, you take T-cells from patients. Let's see if the mouse works here. So this is a patient undergoing an apheresis, a patient with leukemia. You take out the T-cells, you separate out all of the products. You don't want to have any leukemia cells as part of it. You have their T-cells, and basically using a viral vector, you transduce the T-cell to introduce the CAR part of it, which is the antibody recognition properties, to recognize a cell surface marker on a leukemia cell. The vast majority of the experience in CAR T-cell therapy, and certainly what has been, impressive success has been in CD19, where you are targeting CD19 on B-cell leukemia or B-cell lymphomas. And what we are doing is actually doing this with a CD22 and targeting 22. But basically, the T-cells then undergo initial stimulation, transduction, expansion. In the meanwhile, your patient is getting ready. They have active disease, so they might have to wait a little while to get some chemotherapy. But when the T-cells are ready, your patients are undergoing some type of lymphodepleting chemotherapy. This chemotherapy is not really meant to treat the underlying leukemia, although it may have a little bit of a role there, but it's really helping to prepare the body to be in a state to receive these T-cells. So that the T-cells go into the body, they then grow and expand and do what they're meant to do, which is then grow, expand, and target the CD19, or in this case, the CD22 positive leukemia cells. In terms of the current status of CAR T-cells in 2019, so this is really just remarkable to see where we are. The first child was treated with CD19 CAR T-cells in 2012, and sure enough, we now have an FDA-approved construct. So this is the FDA-approved construct for pediatric patients up through the age 25, which is CD19 CAR. The initial data was a phase 1 trial was conducted at the Children's Hospital of Philadelphia, and this shows their overall results, which is their global registration trial. This is based on 75 patients who were treated. It shows an overall high remission induction rate and the durability of remission. You can also see here the impressive overall survival in patients who have relapsed refractory disease. For those of you who are not familiar with these Kaplan-Meier survival curves, this tells you the overall sort of probability of survival, and then this gives you time. And then the other two indications are Kymriah, which is a CD19 approved construct, which is also approved for adults with diffuse large B-cell lymphoma, and Yescarta, which is a different version of a CAR T-cell with a different type of stimulatory domain that makes it equally potent but maybe less durable, is also for adults with diffuse large B-cell lymphoma. And so these are the two approved constructs with two separate indications. So again, this is really exciting, has really helped change the landscape of patients with chemotherapy refractory disease, because up until CAR T-cell therapy, most early phase 1 clinical trials had maybe a 20-30% response rate if you were lucky. And we were seeing response rate, this CAR-based therapy that was 70, 80%, and to have these products now FDA approved has really changed the dynamic. The limitations of current therapy, and that really comes down to this curve right here, which is event-free survival, is that CAR T-cell therapy, while it may have the potential to induce a durable remission, it may have the potential to be curative, and certainly the first patient is in ongoing remission, now 6 years strong. We are seeing relapse, and these relapses range from anywhere between 6 weeks to now 5 years. We got a recent referral that was several years out. And you do see relapses, with about 50% of relapses happening at about 1 year post CD19 CAR. You also have some patients who have refractory disease to CD19 CAR. Also, another limitation, which I'll talk about in the next slide, is patients who can't even get to CD19 CAR. And then also the durability of remission. I think the verdict is still out about who can be cured, and I think currently we don't have the metrics to really, really predict who can be cured or not. In terms of relapse, the majority of relapses are happening with antigen-negative disease. So what that means is that they are losing the CD19 surface antigen on the leukemia cell, and that is really, really hard to be able to predict. You can predict if the CAR T cells start to go away, if your normal B cells start to come back, that they may have impending relapse with CD19 positive disease. But either way, you're left with this 50% relapse rate. And so overall, I like to talk a little bit about what the limitations are to durable CAR T-cell remissions. We will not be talking about all of this in the context of this talk today, but I like to talk about them in 4 separate categories, and there's a recent review article which you can go to after this. But you can either have a patient where you have somebody who's gotten a lot of chemotherapy, you can't even collect their T cells, or you collect their T cells, but you can't manufacture them into CAR T cells for a host of reasons. You can have antigen loss, and this is what I'll be focusing on as we think about CD22 CAR. So you give somebody CAR T cell, and the leukemia outsmarts the therapy by losing the 19, so it comes back. And actually, one of our medical students, Diane Liebert, has a poster actually in this session who's going to talk about what we have seen in terms of 19 evolution in the era of immunotherapy. We have CAR toxicity, so I don't talk a lot in this talk about cytokine release syndrome, but it's important to know what that is. That is a multi-organ system manifestation as a result of the CAR T-cell expansion that creates a flu-like syndrome where you can have just fevers, body aches as a very mild manifestations, and on a more severe spectrum can lead to fatal outcomes, including fatal neurotoxicity, refractory seizures, multi-organ failure. And so we've learned how to optimize therapy, but there are still going to be patients out there who have very severe manifestations. And then this whole area of unmet need, although the CAR T-cell products are FDA approved for lymphoma, we have neither had the experience in pediatric patients nor the outcomes that the adults are seeing with diffuse large B-cell lymphoma, and certainly need to think about how to optimize CAR T-cell therapy for solid tumors. So with that background, I'll go into the rest of the talk, but maybe I'll just ask really briefly if there's any questions before I move forward. Okay. So CD22 CAR T cells, so while CD19 CAR was in evolution and people were actively getting treated, in the pediatric oncology branch, we had developed a long history of targeting CD22. Prior to this, we had used an antibody-based therapy called moxetumomab. Our flow cytometry team really has sort of the world's expertise in looking at CD22. We'd become very familiar with this construct, and so developed a CAR T cell. This was developed by Crystal Makel in her lab, a CAR T cell that specifically targeted CD22. I will say that at the time, our intention wasn't necessarily to only be a salvage option for patients who had failed CD19 negative disease. But sure enough, as you go through time, that's the patient population that we ended up seeing. We had an initial publication based on our initial outcomes. This was a phase 1 clinical trial. We had a standard dose escalation. This is our construct. We treated patients between the ages of 3 and 30. All patients received the same lymphodepletion that you're seeing in a lot of the other CD19 targeted clinical trials. We infused our first patient in 2014, and based on our initial experience, saw that we had about a 70% complete remission rate. And wouldn't you know, the very first 6 patients that we treated were all CD19 negative or had relapsed after CD19-based therapy. So since that time, we have continued to have an ongoing accrual. This represents updated data as of March, but we've continued to enroll at that time. I'll just presenting the updated data that's fully analyzed. But at this point, it was 57 enrolled and 52 infused. We've now enrolled 64 patients and have infused 58 subjects and have maintained the same data that I'll be presenting. So this is 52 subjects. The median age is 18, ranging from 4 to 30. You can tell how refractory refractory this population is. Again, 70% of patients have had a prior transplant. We've even had a few patients who've had 2 prior transplants. Again, the majority have had CD19 CAR or blinatumomab or a combination. Blinatumomab is an antibody targeting CD19, also combined with an antibody ability to target CD3 cells or T cells as well. A good percentage, a little over a quarter of the patients have had prior inotuzumab, and inotuzumab is an anti-CD22 targeted antibody. Some patients, as we went along in the world, have also received prior CD22 CAR T cells, and we quickly learned that it really becomes important in terms of structure and function that a lot of the other CD22 CAR-based strategies were not equally effective, which was very different than the experience with CD19. So we had a few patients who had fell into that category, and then again, 53% of the patients who had any CD19 negative population, and the vast majority had what we call an M2 marrow or a higher burden of disease. We did have 5 patients who were not infused. One we decided electively not to infuse because at that point this patient had a partial population and we did not want to treat that because we'd only effectively treat part of the leukemia cells and she would have all the toxicity without the benefit. We did have 1 manufacturing failure and that patient got treated separately. We had 1 pre-existing condition that was discovered after the event with a patient who had pretty significant radiation-induced CNS necrosis that we were not comfortable treating. We had 1 intercurrent event with an infection, and then 1 patient who at the time was awaiting infusion. The other thing we did that is really interesting in the context of this trial and will be the focus of our follow-up publication is that in the middle of this trial, we tried to optimize the way that we make CAR T cells. And so I mentioned that original slide that sort of shows that little pie chart. One of the biggest issues is being able to manufacture CAR T cells, and we were able to do so even though we had one manufacturing failure. We were, manufacturing CAR T cells for, easily 95% of the population, but then we had 1 patient who presented us with a particular challenge. And the particular challenge was that when you have patients who have a high burden of leukemia cells and a high burden of other cells that can be inhibitory, such as NK cells, it really inhibits your ability to not only manufacture the CAR T cells, but to have the T cells grow and expand and function the way that you want them to. So what we did in the context of this clinical trial is that we changed those dynamics and we used 2 different ways to separate out the T cells. I won't get into the science behind this, but this is sort of a negative selection process where you took out all of the cells that you didn't want after the apheresis, and you were left with a semi-purified product. In this way, we actually used a positive selection where we are actually finding the CD4s and the CD8s, both different components of the T cells, and we selected for the ones that we wanted. So we start off with an even more purified product. And found out that when we did that, we also changed our ability to manufacture the CAR T cell products. We had a much more consistent product despite inherent patient-to-patient variability and also a different toxicity profile. So this is a very, very busy chart, which you don't need to look at all the numbers, but we're going to focus on 2 big areas. So this is a toxicity profile of all of our patients who are treated. You can see here that the patients we were treated, there were 88% who had any grade of cytokine release syndrome. Our cytokine release syndrome, so there is a varying of grades. Grades 1 and grade 2 basically is either low-grade fevers, maybe a little bit of oxygen support, maybe a little bit of blood pressure support, but the vast majority of our cytokine release syndrome in comparison to CD19, it was actually relatively mild, and this did not change with any type of change in manufacturing. This is talking about higher grade toxicity, and this was only a small fraction of the patients, speaking to the fact that there are going to be different toxicities with different CAR-based targets. Tocilizumab is a therapy that we use to help prevent the cytokine release syndrome from being more severe. It helps to block the IL-6 from entering the end organs, and so we used it in about 40% of patients, and we actually started to preemptively use it to prevent even more severe toxicity. So some of this represents both treatment of CRS and prevention of CRS. We did use steroids. We had a symptomatic coagulopathy, so we have seen some elements of coagulopathy. In the vast majority of CD19, it's been asymptomatic, not causing problems, but we had a little bit, again, of a different toxicity profile that told us that different CARs are going to have different toxicities. We had HLH, which is this extended inflammatory response that was seen. And I think part of the problem is that right now this is happening in CD19 CAR, but it is not well defined and explored. And this is something we've learned. So we had to borrow definitions and create our own, so that came up with our own definition. And then again, some other toxicities that we've seen. We did have 2 patients who died on this protocol. One was a direct result of CAR T-cell related toxicity, and one was unfortunately the result of sepsis after the resolution of cytokine release syndrome. But again, telling us that infection and CAR T-cells are a particularly bad combination, and something that we are learning over time as well. And this sort of tells you just sort of sorry, we'll go to that next slide actually. This tells you a little bit about some of the changes. I'm going to walk you through all of these. So this here shows, this tells you all of the different dose levels we explored. And without getting into this each represents like a half log increase in the CAR T-cell dose that we gave. And this is prior to us doing that positive selection. You can see here everybody's sort of fairly uniform. As soon as we do the positive selection, you can see here that we have a much more inflamed response. So this is a ferritin over here on the y-axis. These are the different dose levels. We actually went from dose level 2, which was our active dose, to dose level 2 with the positive selection, and saw that patients were not having a worse cytokine release syndrome, but that they were just overall more inflamed. We were having to use more steroids. And then we ended up actually going down a dose level. And this was really interesting because prior to this dose level was almost essentially ineffective. We had treated 6 patients at this dose level and only had 1 responder. And this is currently the active dose level where we have about an 88 or 89% complete remission rate. And the only single difference in the context of this clinical trial was the way that we took their initial product without actually making a change in the manufacturing. This is broken down now by looking at all the patients who had the negative selection, which is a T-cell selection, versus a more positive T-cell selection. You can see here that everybody has an overall more inflammatory response. This is looking at IL-1B, which is one of the metrics of the HLH, which is, again, this multi-organ system manifestation. And again, this is our biggest problem. Once we did that positive selection, we saw patients who had a lot more elevated markers. And to give you a sense, so IL-1B, it should actually never be able to be detected. It's such a fleeting cytokine. And we are getting levels in like the 40 range. That tells us, how inflamed these patients were able to get, and we're able to treat them down. Here you see IL-6, and again, just sort of without going through all of these, you can see here this tremendous jump that we took going from dose level 2 to dose level 2 with the T-cell selection. And then again, we brought everybody down to a little bit more manageable profile, though there's a few patients that are still quite inflamed, and the changes that happen over time, and also with IL-10 as another marker of that inflammatory response. So what we learned in the context of the CD22 CAR T-cell cytokine release profile is that we saw capillary leak syndrome, which again was something that had not been talked about much with CD19, but this was something that was unique, that seemed out of proportion to the degree of cytokine release syndrome we had overall, and was actually unique to, it was interesting, in our population, we had previously had some experience with the CD22 targeted antibody, where we saw capillary leak and we saw hemolytic uremic syndrome, which again, I'm mentioning all of these different inflammatory things, but these were things that were seen in the context of our clinical trial that really had not been reported on with CD19. And so one of these was that CAR-related toxicity. We did actually have to treat 2 patients for hemolytic uremic syndrome, which again is a manifestation of kidney injury, endothelial activation, and hypertension, along with some thrombocytopenia. We did have to come up with a definition, but at least we have a definition that we used that was able to help identify this toxicity. We saw this symptomatic coagulopathy that was essentially resolved when we went back down to a dose level. In our neurotoxicity, so this was really interesting. So we now have a CAR T-cell trial that is equally effective as it compared to CD19, but we have nowhere near the neurotoxicity seen on CD19. We have a couple of different things that are going on, but we don't have really any neurotoxicity. We had no seizures, no aphasia. Everything that we had was generally mild. I think we had maybe 2 patients that we've given steroids to for some neurotoxicity, which again tells us that there is something different about this CAR and the toxicity profile where you can have endothelial activation, but it may manifest in different ways. This is our overall complete remission rate based on 51. Like I said, we've now treated 58 patients, and it's essentially the same. We had a few patients who had progressive disease. This poor patient here, who is the only patient at dose level 2 who has progressive disease, basically had very extensive disease progression even before the CAR T cells had a chance to work. So I had to give this poor patient steroids and chemotherapy essentially on day 4 after the CAR T cells were given, simply because the white blood cell count went too high and we could not have a chance to let the CAR T cells work. But everybody else here, you can tell overall this goes through the various dose levels, but if you focus really on dose level 1, dose level 2, and dose level 2 without the T cell selection, we have an overall complete remission rate that is quite respectable of all comers. If you look at those that were beyond that initial experience, we actually have an 80% complete remission rate, and the vast majority of patients who achieve a complete remission are MRD negative or minimal residual disease negative. In terms of CAR T cell therapy, so we found that it can effectively traffic and eradicate extramedullary disease. So in this case, we had a patient here who has this lymphomatous mass pre-CAR, and by day 28, it's completely resolved. This is a PET scan. This is another patient who has extensive bone marrow disease, but also has a bunch of lymphadenopathy. This is a patient who is CD19 negative, and you can see here that this is a pre-treatment. He has a very heavily involved bone marrow. At 1 month, he achieves complete MRD negativity, but there's still some residual activity going on. It takes almost a good 6 months. And we also see the CAR T cells are continuing to be there, continuing to provide an anti-leukemia effect until he becomes completely MRD negative, so that there may be a little bit of a delay in the optimal response for lymphomatous disease as it relates to marrow disease. We found that the relapse was associated with modulation of CD22 expression. So similar to the whole idea behind CD19 negativity, we have the problem with CD22 as well. And so with CD22, not only can you lose CD22 and have the leukemia relapse, but even simply sort of going beyond a threshold will be enough to have you relapse with CD22 dim disease. So these are a few patients. You can see this is their site density. So we quantify the sites on a leukemic cell and this is a logarithmic scale, so you can tell how much CD22 expression is. This is prior CD22 CAR, and you can see the changes that happen for those who relapse after CD22 CAR. The median time to relapse was 168 days, so the majority do have relapse. We have a few patients who have prolonged complete remission, but we have relapse now. Overall it's about 6 months. The median overall survival is just about a year, and we have one ongoing complete remission that is more than 3 years. We have another patient who is out getting to be close to 2 years, where he did relapse about at 1 year after CD22 CAR, relapsed with CD22 positive disease, got a single infusion, and remains in an ongoing remission with just that therapy. And then we have been using this as a bridge to transplant. So I think as the dynamics change, transplant has an essential role in patients with relapsed refractory leukemia. And given the dynamics of CD22 and the really refractory patient population that we get, which is majority having failed CD19 CAR or blinatumomab or chemotherapy, we think that their disease also may have progressed to becoming even more refractory, so we're using this as a bridge to transplant. We found that prior CD22-directed therapy does not preclude a response. So you can get inotuzumab, you can get a CD22 CAR therapy elsewhere, but it may make our response a little bit less likely or less durable. So we are looking at prior inotuzumab, and we have seen it lead to partial CD22, and we don't like to treat those patients. We are still able to effectively treat those patients. You can see their site density is lower overall with those who've gotten prior CD22 directed versus those who didn't. You see that the MRD negative remission rates is 93% in those who have not gotten prior CD22 targeted therapy versus 67%, and then the remission durability is shorter. So I think it sort of sets them up to relapse more quickly. And then basically our overall summary is that the extended experience confirms the efficacy while providing insights into unique toxicity profile and the impact that manufacturing changes can have. CRS grade was generally less severe than CD19 CAR, but we learned some unique aspects of CD22. Despite the evidence of coagulopathy and endothelial activation, neurotoxicity was mild. I talked already about the changes in the inflammatory response with a minor modification to just the selection. And then we found that the impact of prior CD22 targeting can impact treatment response. So it is the largest study to date of CD22 targeted CAR T cells. We have treated 58 patients. It's really the most active clinical trial. We have a favorable response and acceptable toxicity profile. It is a salvage for patients with relapse or non-response to CD19 targeted therapies. It can be a potential as a bridge to transplant, and we're currently working on writing a phase 2 clinical trial with the phase 1 continuing to have ongoing enrollment. So many people to thank, but mostly thank you all for letting me have the opportunity to speak. Thanks to all of my colleagues here who have referred your patients for this clinical trial. It's really quite an experience to have a phase 1 trial that has so far exceeded its enrollment, to be enrolling 60-some patients right now. We plan to close the trial, but there's an ongoing clinical need, so we'll continue to treat. That's all.